So the discussion is about the glycosidic bonds or glycosidic linkages. Especially we focus on the alpha 1,4 glycosidic linkages and beta 1,4 glycosidic linkages. Why we are going to discuss about these different types of alpha and beta bonds means the particular enzyme can break or digest particular bond only. For example, amylase, that is actually called as alpha amylase, can break the alpha 1,4 glycosidic bonds only. But beta glycosidic bond or linkage are broken by cellulase enzyme or beta galactosidase enzyme. That is our discussion. So we have to discuss about the different types of glycosidic bonds. These are called the glycosidic bonds or linkages. In this glycosidic bonds, we focus on alpha 1,4 glycosidic bonds and beta 1,4 glycosidic bonds and linkages. First, what are our food materials? We will see. If we eat plants, we can get starch. Starch is containing two types of amyloses. Those are called amylose and amylopectin. The amylase is a linear branch, one glucose molecule, one another glucose molecule, linear chain. But whereas in the amylopectin, the chain is branched like this. So many branches are there. In these branches, we can see alpha 1,6 glycosidic bonds also. Then another one is we will discuss alpha 1,6 glycosidic bonds also we will discuss at the same time. Next, disaccharides, maltose and sucrose. Maltose is made up of two glucose molecules. Sucrose is made up of one glucose and one fructose molecule. So these are our food materials. And if you will eat animal source of food, that is we can get the polysaccharide is the glycogen. These are the polysaccharides in plants. These are the polysaccharides in the glycogen in animals. In this glycogen is also a linear polysaccharide which is made up of all monosaccharides called the glucose. These all are digested by alpha amylase enzyme. Why these all are digested by alpha amylase enzyme? Because we will discuss now. Next, in the plant source of food, another type of abundant polysaccharide is there, that is called cellulose. Actually, the cellulose is abundant in the plant source of food because all the cell walls in the plants are made up of cellulose. Those animals which eat these plants can get the cellulose, especially for example ruminants, cow, buffalo and some other animals depend on cellulose digestion only because they need lot of energy, but they eat only plants. Sometimes buffalo and cow can eat only the grass. From that grass or from that plant they have to get lot of energy. But this cellulose is made up of beta 1,4 glycosidic bonds. These are made up of alpha 1,4 glycosidic bonds. The cellulose is digested by cellulase enzyme only. Cellulase enzyme only. In our body there is no cellulase enzyme. No one gland in our body can release the cellulase enzyme. Then we do not digest the cellulose food material. Another one is lactose. Lactose is also made up of beta 1,4 glycosidic linkages. This is this is called the milk sugar. In infants, this lactose is digested because the beta galactosidase enzyme is present in the infants, but in adult human beings there is no beta galactosidase enzyme to digest the milk. So lactose. This is disaccharide which is made up of glucose and galactose. So these are our food materials. Some are disaccharides and some are polysaccharides. Okay. Now what are alpha 1,4-glycosidic linkages and what are beta 1,4-glycosidic linkages and what are alpha 1,6-glycosidic linkages? These alpha 1,4-glycosidic linkages. This is sugar molecule, that is hexose sugar. In this hexose sugar, in a polysaccharide, one glucose molecule or one monosaccharide covalently bond with the next monosaccharide. Next. That covalent bond is called the glycosidic bond. This is one glucose molecule and it can covalently bonded with another glucose molecule side, nearby glucose molecule. So in this is the sixth carbon, this is first carbon C1, second carbon, third, fourth, fifth, and this is sixth carbon. This is hexose sugar. In this sixth carbon, this is first, second, third, fourth, fifth carbon, 5 carbons, and this is the sixth carbon. So this is hexose sugar. In between these two hexose sugars or monosaccharides, normally the glucose molecules, the bond should be formed, the covalent bond should be formed. That covalent bond is called the glycosidic bond. Whether the beta bond is forming or the alpha bond is forming, we have to discuss now. How do the alpha bond is forming? How do the beta bond is forming? Here, this is called hydroxymethyl group. This is hydroxymethyl group. And the important thing which we have to discuss is this first carbon. This is anomeric carbon. What is this anomeric carbon? The carbon which is involving in the formation of covalent bond, the glycosidic bond, are called the anomeric carbon. This is first carbon. First carbon is covalently bonding with the fourth carbon of the neighboring glucose molecule. Here, how can we identify whether it is alpha bond or beta bond? Now you can see this is hydroxyl group in the first sugar. In the neighboring sugar, this is hydroxyl group. In between these two hydroxyl groups, the bond is forming, the covalent bond or the glycosidic bond is forming in between these two hydroxyl groups. Where is the hydroxyl group to the hydroxymethyl group? The hydroxymethyl group is at the top region of the sugar molecule and the hydroxyl group is located below the anomeric carbon. So this hydroxyl group is opposite to the hydroxymethyl group. These two are opposite. This is called alpha. So this is called alpha carbon. If it is like this, that is called the alpha carbon. So here what is happening during the bond formation? When the bond is formed, normally during the photosynthesis, the glucose molecules are synthesized. The synthesized molecules, glucose molecules or monosaccharides can form the glycosidic bonds. Here, during the formation of a bond, a covalent bond, one water molecule is removed from this. In this, one H2O, one water molecule is removed, what happens? The bond is formed like this. One water molecule is removed, H2O is removed. With one oxygen, the bond is forming, the covalent bond or glycosidic bond. What type of bond this is? This is alpha glycosidic bond or alpha 1,4 glycosidic bond, because 1 to 4 the bond is forming. This is called the alpha 1,4 glycosidic bond. This alpha 1,4 glycosidic bond is broken by alpha amylase enzyme present in our saliva pancreatic juice and succus entericus of small intestine only. Next, alpha 1 4 beta 1 4 glycosidic bond. Sugar molecule, for example, we will take cellulose, a polysaccharide. And neighboring hexose sugar. Same methyl groups. I am writing methyl groups and the hydroxyl groups. Hydroxyl group is located here. That is same side of the hydroxy methyl group. The hydroxyl group in the first sugar is to the same side of the hydroxy methyl group. But whereas in this alpha bond, alpha sugar, this hydroxyl group is located at the opposite region. That's why it is called as this carbon is called alpha carbon. So here OH group is there. Then in the neighboring hexose sugar or glucose, the OH bond is located. OH group is located here, hydroxyl group. So here what happened? During the formation of covalent bond or glycosidic bond, the bond is forming like this. One water molecule is removed. Water molecule is removed and the bond is formed. How is this bond? The bond is like this. This is oxygen. The bond is forming like this. Here the bond is like this. This is alpha bond. If the bond is like this, this is beta bond. This beta bond is not broken by alpha amylase. This is broken by only the cellulase enzyme and the beta galactosidase enzyme. Beta galactosidase enzyme. So that is the difference between alpha 1,4 glycosidic linkage and the beta 1,4 glycosidic linkage. Then another one is, here we have all discussed amylopectin. In this amylopectin, glucose molecule and glucose molecule. In this cellulose and in the amylose, the glucose molecules are linearly arranged. There are no branches. But whereas in the amylopectin, we can find some branches like this. So many chains. So many chains we can see. So the structure of this amylopectin, we will see glucose molecule, hexose sugar, another glucose molecule, same like that. In the branch, for example, this one branch. So, how is the bond is forming in between the sugar molecules? If they are in branched, for example, amylopectin. In this branch, first carbon, second, third, fourth, fifth and sixth hydroxymethyl group. In this case, first carbon, second, third, fourth, fifth and sixth. This is also hydroxymethyl group. Here hydroxyl group is there. This is the linear branch and this is the side chain. In the side chain, the hydroxyl group is there. While the covalent bond is forming, the glycosidic bond is forming, the alpha glycosidic bond is forming, one water molecule is removed as usual. And the bond is forming here in this bond. The bond is forming in between first carbon of the previous sugar and the sixth carbon of the next sugar. And this is oxygen. So here the bond is formed from alpha. This is alpha because the hydroxyl group is opposite to the hydroxymethyl group. 1, 2, 6 glycosidic bond. These bonds are only found in the aminopectin. So the final conclusion is alpha 1,4 glycosidic bonds, alpha beta 1,4 glycosidic bonds, beta 1,4 glycosidic bonds are present in the cellulose, which is digested by the cellulase enzyme. In the ruminants, some of the bacteria live in the small intestine of the ruminants. Buffalo, cow can release the cellulase enzyme which can digest the food material. Actually, the ruminants themselves do not release the cellulase enzyme, but the bacteria which lives inside the small intestine or the stomach can prepare the cellulose enzyme and that can be used to digest the cellulose food material. Those bacteria are called the symbiotic bacteria.